recording or?
Yes, we're recording.
We are recording. Excellent. Okay. So the material that Herbert covered was about constructing models of different kinetics, and there are a lot of great questions, which I think it's fascinating about, embedding these details of the chemical interactions into, more complex communities, whether they're, individual cells or larger ecosystems, and to see how that all interplays. But as you know, from looking at those kinetics, there are a lot of parameters. I mean, just the kinetics constants, or they may be, maximum reaction rates, like in Michaelis-Menten, that we have to somehow incorporate. And we don't know these a priori, and we may have some estimates from experiments that are done under conditions that are typically not physiological, but somehow we have to come up with these constants, and there are a lot of them. So that's what this is really about, this notion of parameter fitting. Out of curiosity, I don't know, maybe one or two people can speak up. How many of you folks are already familiar with the idea of parameter fitting, have dealt with this in some of the work you've done? Anybody offer something? Okay, I see a thumbs up there. Okay. I mean, this is where, the models themselves, especially when you talk about interacting models, like, an ecosystem of microbes interacting, that is computational intensive, that can be. But now if we throw on top of this notion we have to estimate the parameters, it can be really computationally intensive. And this is actually one of the core challenges we face in the community in terms of doing these kinds of simulation models. So what I want to do is sort of take you through this. I'm going to focus on relatively simple systems, like basically it's a single cell is going to be an environment I'm primarily looking at, that you sort of get a feeling for. We encounter a lot of challenges here just with even modest sized pathways. So it's a small step to see how those challenges then multiply when you get to larger communities. Okay, so let me move along here a little bit. Okay, so for those of you who have not encountered this concept before, my running example here is going to be glycolysis, and there's a particular model that was developed by Jenna Wolf in about the year 2000. It's a nice model because it's relatively simple, and we're talking about glycolysis, which that's probably the first biochemical pathway you studied. So everything seems to be sort of under control in terms of, wow, we should do a great job with simulating this. And so the challenges that arise are actually rather stunning. So here's a brief abstract of this, the article point. It's actually one of the, I don't know if you've heard references to biomodels. It's a repository of models. I think most of them are many of them are SBML, and you can they're sort of like ready to run. They've been curated, which is really nice. If you look at this model is about, you see the key thing is that it depends on the behavior of the model depends critically on these parameters, on these kinetic constants. So if we look at the model itself, and then, if you choose the right constants, you can get agreement with the data, which, right away, you should be a little bit concerned about, are we actually fitting the data are we actually fitting the parameters to the data and sort of getting conclusions that we like, rather than being scientists and actually figuring out whether or not we have a credible hypothesis that we're testing. So here's the model. It's a little bit on the small side, but you folks are all on Zoom, and so you can read it more carefully. This is actually a bit of a translation of the original model. The original model was not written using, sort of chemically interpretable names. So you actually see glucose over here, and ATP is recognizable. I think they used the original model. This was S5, which is not so understandable. Fructose 1,6-bisphosphate, also very recognizable. So you all, all the normal players you expect in glycolysis, with the you have the investment and then the return phase of glycolysis, and you see about 10 reactions going on here. The key thing we're going to focus on here are these constants. Like here's J1 underscore K1, which is a constant here for the kinetics over here. And this is written in antimony. You should be familiar with at this point. If you want to look at the kinetics, we've got the mass transfer part in the beginning of the reaction over here. So you see, we've got glucose and 2 ATPs going to fructose 1,6-bisphosphate. And we get 2 ADPs, so we consumed a couple of ATPs, got some energy consumption going on there. But here's the kinetics that were used in that particular model. You should be familiar with this, just look for after the semicolon. What we're going to be focusing on are all the constants. So there are, what we got here, about 16 constants. And this is just glycolysis. It's a relatively simple model. So you can see where this is what they got. They were showing that under the right conditions with the right population, you can end up with oscillations. And so that was a sort of interesting mathematical analysis, and they got some correspondence to some experimental data. But then if you go back and look at the model, all these constants here, that all these areas of the model that are in these boxes here, these are all the constants that are present. And there are a lot of them. They're just sort of sprinkled throughout. Some of these we may have some initial estimates of, some of we don't have initial estimates of. And so how do we figure this stuff out? And that's really what this section of the course is about. And this is a fundamental part of the workflow you need to go through to really be a researcher doing this kind of modeling. Just a second, we've got a little bit of chat. Just trying to see if there's anybody who, okay, fine. I think those are side conversations. All right. Oh, Veronica, could you do me a favor? If somebody has a question, especially a question where there's a lack of understanding of something I'm saying, could you signal to me? You have carte blanche to do that.
Okay.
Thank you. Yeah, thanks. Okay, so let's talk about what this fitting stuff is about. I think conceptually you got the idea of what we are trying to do. What we'll have at hand is we'll have a model that we've developed, and that model will have a whole bunch of unknown constants. We call them parameters. Most of the time, they're going to be kinetics parameters. Let's go back here just for a second. You can see the kinds of things that you've got here. So here we have an inflow rate. Now, this is almost certainly something that we're controlling the experimental environment. So I'm a little less concerned about that one. That we could play with a little bit. This one looks like a kinetics constant over here. Look at this one over here. I assume that Herbert took you through Hill equations. This is actually the exponent. We've got a concentration here of ATP. We're dividing by something that looks like a kind of kinetics constant, and we're taking this whole thing to a power. So those are really the three most common kinds of roles that constants will play. Needless to say, this will ultimately become a system of nonlinear ordinary differential equations. And so this whole system here, needless to say, is nonlinear, and the constants participate in the characteristic of the nonlinearity. So this is a non-trivial system to solve for those constants. So let's talk about how we formalize this whole process. So the objective is we want to find values of these parameters that makes it so that the model produces results that are close to the empirical data. So, in the running example here, we've got about 16 constants. If you ignore that, input rate that's coming in, that boundary reaction, 17 including that. And then the core idea here, and this is one I am very curious to hear how people have encountered this before, are the notion of residuals. How many folks have heard that word before? Just a raise of hands, a nod, a voice. Trying to see how many, and just sort of see where we are. Residuals, okay, Kyle, thanks. Anybody else besides Kyle? Okay.
Yeah.
Okay, a few. Okay, excellent, Megan. Okay, quite a few of you, Rebecca. Okay, excellent. Okay, so a few of you have. So this is a fundamental idea that originates in statistics. And the concept here is that we have observations that we've taken presumably from running measurements, from running experiments. And then we have a model. And the model produces predictions of what should happen under the conditions of the experiment. And residuals, what they are, that's the difference between those observations and our predictions. So if the residuals are 0, we're spot on. Of course, I wouldn't even be talking to you if that happened a lot, like if it almost ever happens. So almost certainly the residuals are not 0. So if the residuals are positive, then our estimate is too small, and if the residuals are negative, our estimates are too large. And it's around sort of managing the residuals. That's where all the work goes in, how we sort of adjust our model. The other thing to notice here is we're not talking about a single predicted value. We're talking about values over time. So it's sometimes during the course of an experiment, we may see a larger value or a smaller value for something like concentration of ATP, for example. And the other thing is that we have many floating concentrations that we're interested in. Mostly, I'm just going to talk about floating species that, whose concentrations vary over time. They're not controlled by an experimental environment. But the other thing that someone could consider is the chemical fluxes of reactions. That might be something else. But in this discussion, I'm just going to focus on floating species. So that's sort of core concept here is this notion of residuals. So how would we in general sort of approach this problem? And really it's a kind of optimization. If you've encountered things like gradient descent before, that's an example of an optimization algorithm. We use differential evolution a lot, but the idea is you want to try and find the best set of parameter values, and that will be reflected in the nature of the residuals. The residuals should be small in absolute value. So if you're talking about how, in general, an algorithm is going to work for doing this fitting, we're going to have a model like, the kind of model that Herbert has been talking with you about. You're going to, come up with a set of reactions. The reactions have kinetics. This ultimately forms one of these system of nonlinear differential equations. You're going to have parameters that you want to estimate. So there may be some parameters that you feel like, gosh, I really know this. I've done this with other studies, whatever. And then there are going to be some subset of the parameters that you need to estimate. We started out with like 16 parameters for the Wolf model. I don't know how many of those we feel confident about. Maybe we might be able to get away with a few that we don't estimate, but probably we're going to have at least a dozen we have to estimate. And then what we have is the observed data. Now in an ideal world, we would have observations of all chemical species. But the reality is for the experimental environments, we typically don't have that. We have a subset of those values that the chemical species we can actually see. And the output from this is going to be, good parameter estimates. And so, like I said before, the idea is it's a kind of optimization where good means that our residuals are small in absolute value. And here's sort of an idea of, sort of intuitively how such an algorithm might work. So until you find a good fit, whatever good fit means, because that's a little subjective and you have a little bit of leeway around that, but typically what it means, good means that if we look at the residuals and we square each residual value and take that sum of squares, so you hear this term, sum of squares of the residuals, that's a single number. And we're trying to make that single number as small as possible. Obviously, it's a positive or it's non-negative because you're talking about squares of real numbers. So the sum of squares of the residuals is typically going to be our measure of, something be the relative quality. So until you get a good fit of the observed values, or you're running too long, and I'll get to that one in just a second, because this can take a long time, even for modest sized models, then here's what you do. Okay, here are the steps. So the first step is you're going to get a new assignment of the values to the parameter estimate. So somehow we're picking new values for parameters. How do we do that? And that's really the core of sort of what we call the search algorithm. So I'll delay that discussion right now. So we can come up with new parameter estimates. Then once we have parameters, now we can do simulations. And you've just been looking at simulations in depth. Once we have simulation results, we can get down here to step 3, and we can take the difference between the observed values, that's data being supplied, remember we've got the observed values up here, and the simulation results. So now we're back to residuals. Okay, and then we can look at the residuals. If the residuals are smaller than previously, and by smaller I mean in terms of the sum of square criteria, for example, then hey, these look like a better set of parameter estimates. Let's use those. Let's use those new parameter estimates. And if the residuals are very small, then we may decide we're done. You know, maybe the residuals are so small, it doesn't look like they're changing. Now we're done. If we aren't done, we continue this loop, and we do that until we exceed a time limit. So that's sort of the basics. Let me pause it here for a second to see if this sort of conceptual framework makes sense before I get into some more details. Any questions at this point? I see there's some things in chat. Okay. Do the data always need to be time course? Okay. No, I'm going to focus on ODEs, but the data don't have to be time course. I mean, for example, even with an ODE system that you're looking at, you could have the data be just the steady state values. Now, obviously, that means that you're only trying to fit a part of the the system's response, just a steady state response, but certainly it could just be steady state values. But then, of course, you're not necessarily fitting to the dynamics. Okay, and I see Veronica already answered that. Okay, let me move on here. Then that's the search algorithm. You will hear a lot of discussion of the choice of the search algorithm, and it does make a difference. But I think to really understand what's going on, you have to sort of take a step further back to get an appreciation for what it is that you're doing and sort of the magnitude of the challenge technically of doing this search at all, regardless. Well, there's one exception. So if you have a nicely structured search space is what it's called. You know, when you're looking for the parameters, if it's nicely structured, something called convex, then you're in luck. But I'll tell you, your chances of finding that lucky search space are much worse than winning the lottery. So, you know, I wouldn't count on it. Okay, so here's one concern. We're fitting a large number of parameters, typically to a small data set. So how much experimental data do we really have? And typically it's not a lot, you know, a few dozen points, maybe a few hundred points. We've got, you know, as I'll show you shortly, even with this glycolysis model with like 16 parameters, once you consider the number of different values of those parameters you want to look at, that data of 20 or 100 points is actually really small. And so there's a concern here. This is a famous quote from John von Neumann, is that, you know, if I have 4 parameters, I can fit an elephant, and with 5, I can make his trunk wiggle. So it's sort of a funny way of saying that the more parameters you add, yeah, I've got more degrees of freedom, but then am I really saying something, or am I just fitting my model, you know, make it, you know, big enough so that I can, you know, account for all the data? So that's one concern. And we have to be mindful of that. There are techniques that are commonly used, something called cross-validation, for example, which I'll touch on briefly tomorrow, but we don't really have the time to go through it in detail, that deal with this. If you've heard if you've done any work with machine learning and heard the term generalization error, that really touches on the same idea. Cross-validation is one technique that's commonly used in that space. So that's certainly a concern, but I think we have ways of addressing that. Okay, and the observation, of course, I mean, as I showed you for the simple, you know, glycolysis model, 5 parameters is nothing. I mean, 40 is more common. And what we're dealing with in this model is 16. Okay, here's the second concern. This has to do with the computer science term is called computational complexity. That means, you know, how long is it going to take to actually do this calculation? So a great way of looking at this is to do sort of, you know, a very simple example. And I'm going to take through this notion of the, you know, sort of a simple assumption that we've got a simulation, and what we're going to do is to just talk about the time it takes to run the simulation. And so suppose that each run of the simulation takes a microsecond. Now, that's probably a pretty simple model. And that's, it takes a microsecond, but of course I have to have had assigned values to all of the parameters of the model. So like in the Jenner-Wolf model, I would have had to assign values to each of the 17 parameters. And what I'm trying to do is find, the right value assignments, or what I call settings, for the parameters so that I get a good match with the experimental data. So I have to do this a lot. Okay, so what I'm going to calculate here, because these numbers are going to get big, I'm going to talk about the units for the runtime of the simulation are ultimately going to be not just in hours, it's going to be in the log base 10 of hours. So, a 1 hour run, you'll see a value of 0 because it's a log base 10. A 10-hour run, you'll see a value of 1. So what does it take to simulate all combinations of parameter settings? So we're doing an exhaustive search is the term here. And so if we really want to find something, we have to sort of search this big space. And this is, what it's going to take. Here's the, over here is a scale I'm going to be using here. It's easier just to color code this. So, here we see that if it's the runs for an hour, you have a 0. If it's a second, it's a negative value because we're talking about logarithms. So this is a logarithm of fraction. Here we go, a day, a month, a year, a century, age of the universe, which about 14 billion years. Okay, so you can see where that's going. You can probably already paint the picture yourself. Okay, so here's what we got here. Let me explain this graph. So we've got the number of parameters we're looking at across the top here. These are the columns. So with more parameters, we have a larger space to search. And over here, around the rows, we got the number of settings. So, we're considering 2 values for the parameters, 4, 8, 16, and so on, up to 64. Now, the first observation is, with the Jenner-Wolf model, we're somewhere over here. That's about where we are. I don't know many biochemical models that are over here, but, there are probably some simple ones. And it's very common for us to be over here. And the number of parameter settings, these are continuous valued parameters. So, we sort of have to explore quite a bit. The observation is if you're over here, yeah, it's, it doesn't take too long. But once you start moving over into this region, certainly with more parameters and more settings, we're way past the time it takes to, anywhere remotely feasible that could be calculated. Now, in some sense, this is sort of an extreme pessimistic view because it's assuming we're searching this entire space. And you could make the statement, well, gee, that's pretty dumb. Do I really have to search the whole space? And that's true. There are some cases where you don't, and I'll get to that in just a second. The problem is we don't really know what this space looks like, and we can come up with heuristics. And we'll talk a little bit about differential evolution. I mean, that's a great heuristic, but it's not guaranteed to always work. And so you're sort of like, you spun the dice, and sometimes it works, sometimes it doesn't. Okay, so let's talk about, yes, go ahead.
Comment or a question in from the chat, just talking about parameter sensitivity analysis before doing fitting. And how would that help?
Oh, yes, right. Okay. Yeah. So parameter sensitivity analysis is yet another consideration. So you sort of layer these considerations one on top of the other. And so if you have found a set of parameters that give you good results, the question is, how many of them really matter? Do I have a parameter value that maybe my model even is particularly dependent on? If I find that I can vary that parameter value a lot, and I still get good results, then it's probably not an important parameter. Maybe I even need to simplify my model or get more data so I can be more responsive to it. Whoever offered that question, hopefully that is a sufficient answer for you. But that's actually an addition beyond this. So,
could it be helpful
okay, so let's talk.
Could it be helpful beforehand to sort of say, like, if this parameter is not very sensitive, then I don't want to estimate it, because although I'll spend a lot of time like searching all the different values and it doesn't make any difference.
Yeah, I mean, that's certainly true. I think, Lucian, and help me with your thinking there. The challenge, I think, is how do you know that it's not sensitive to that parameter?
In like some other setting, like if you change some other parameter,
Okay,
now
right. If you
suddenly
knew a
your first
priori from
parameter
some other
is
setting,
sensitive.
yes,
Yeah.
right. And then what I would strongly recommend is, and this is the argument for start simple, right?
Yeah.
Don't start with a complicated model that throws everything in. Start simple. That's how we're doing on time. I think we're doing okay. So I give you a brief story, and I apologize. And actually, tell anybody you like, Lucian or Herbert or Veronica, if you don't like my stories, and I'll stop. But in my previous life, I'm a computer scientist by training. Much of my work has been mathematical models of large computer systems. I was at Google and Microsoft and IBM, and so a lot of my job ended up being trying to convince software developers of large systems that modeling was a useful thing to do, that we could predict, the computational resources, the electrical power, whatever it was, that this was a useful thing to do. And a lot of times it boiled down to someone saying, well, wait a second, your model doesn't include this locking architecture that I use inside the operating system. I said, yeah, it doesn't, because I don't have to consider that to predict performance. But there was a lot of skepticism of the model because it was not sufficiently detailed. And so this is part of the art of modeling. And what Lucian, I think, was referring to is that you only include those details that actually make a difference for what it is you're trying to predict. The other details should be removed. Otherwise, the model's so complicated that you're really just doing measurement. You're not doing modeling. So anyway, hopefully that story is helpful. If not, I apologize. Okay, so let's talk a little bit about this challenge of the surface that what I refer to as a surface. I'll actually do some graphic here. Okay, well, I got some positive feedback. I tell my kids, I have 2 adult children. I tell my kids who are, gee, that's another dad joke. I said, look, if you don't like it, fine, but I really enjoyed it. So hey, that's the way it goes. Okay. So fitting surface, what is a fitting surface? So I'm going to talk about a very simple model for right now, one with just 2 parameters. So here's my model. I'm producing X, some species X, and then I am consuming it. And so it's based, I've got this the simple model here for the production. This is all mass action. Probably heard that from Herbert. So it depends on the concentration of this initial species over here. I've got this unknown constant K1, and then the depletion of X or degradation of X is based on this the amount of X itself, and that's a constant K2. And so this is going to be the rate of change in X. Okay, very, very simple model. So let's take a look at what a fitting space actually looks like. So here's what happens if you do the simulation of this over many different values of K1, K2. And what you'll see is something like this. So what I'm calculating here are the residuals, actually what's called so I'm taking the sum of squares of the residuals, and some people will do the square root. I'm actually doing the square root as well. That's what, if you run into the acronym RMSE, the root mean squared error. That's what that is. It's the sum of the square of the residuals, and you take the square root. And I've got a heat map over here where the lightish gray over here is 0. So that's where we'd like to be. We'd like to be at 0 because that's where, we've matched the data. So I generated this data. I think the true values here for K1 and K2 are 1 and 2. And so what I'm doing here is I've got that's the real model. And then I'm looking what happens as I change the values of K1 and K2. What does it look like in terms of the heat map? How large do the residuals get? So I know where I should go. So if you look at this is what the true values are. And as I move away from this, I'm over here. So if I had a search algorithm that started, let's say, out over here, it'd want to move towards this area, which is lighter and lighter and lighter. And actually, if you look at this, if you get close to this, let's do a close-up of the surface. So now what I'm doing is I'm doing a close-up over here where I'm doing just looking at these values that are closer by, you can see, and so the scale has changed too. Now the dark is 0.135, where it was before it was like 2.7, because I'm doing this close up. But you can see there's a noticeable light area right here along and through here. And that's because this surface has a what's called convex shape to it. That is, if I have an algorithm that somehow could read this color, and it just basically moved away from the dark areas, going to the lighter areas, I would probably end up right around that red dot, which is the true value. That's where I want to be. This is an example of what's called a convex fitting surface. And of course, the algorithm wouldn't read the color. What it would do is it would look at the RMSE values. These are large, and so it'd start moving this way, and then say, gee, this is smaller and smaller, and eventually end up with the red dot. This is the kind of fitting surface that scales really well. You can get, a large number of parameters if you have a convex fitting surface. And then your life is good, and definitely you should play the lottery if you find you're hitting a lot of convex fitting surfaces. So let's go back to reality, away from convex fitting surfaces, and look at another example. And okay, and this is the other idea, it's sort of another intuition. This is actually sort of a roadmap for how you find what are good parameter values. So this would be our values for K1 and K2. Okay. Oh, one more point here. I just wanted to make sense. I talked a little bit about that generic algorithm, about this notion of gradient descent. And you can see that's actually very graphical and intuitive here, because I've got this gradient of RMSE values, and I'm going down that gradient towards the true value. And that's what gradient descent does. And for a convex surface, that's exactly what you want to do. I mean, actually, one of the key properties, mathematical properties of a convex surface is that the local minima, so if you just move locally, and that's what you're doing, you're moving locally through the space, is the global minima. And that's a very powerful characteristic, that you can actually find the global minima, the best value, just by moving locally. And that's what a convex surface gives you, and what gradient descent will do for you. Okay, now let's go back to the Wolf model. So the Wolf model is a little bit different. I'm just going to consider two of the parameters to be fair. So we're in the same kind of framework as before. It's a more complicated model. You remember there's a lot of nonlinearities in it. I'm looking at two of the parameters. They were earlier. I'm not going to, I don't show the reactions here. I probably should, but these is for these two parameters, J2 underscore lowercase k and J1 underscore capital Ki. And I, like before, these are two parameters. I put them on this surface over here. I show you what the true value is as, that model was published. True in the sense that this is the one that was published. So that's the red dot that we're looking for. And you can see that our fitting surface looks a lot different than that neat little convex surface. Do you see all these areas in through here where it gets dark and then light? Almost looks like ridges on a mountain. And actually the analogy to a mountain is very apropos, because it's much like you're walking through a mountain range, and you can find yourself in a valley and think, aha, I use gradient descent, and now I have the best fit to the parameter values. But you're just in a local valley. You're just like somewhere over here in this local valley. And if you just walked up this hill, you'd find there was a deeper valley, and I'd have a better value. Or if you walked up this hill, and so on, until I got to the one which is actually the true minimum. And that's the challenge with doing fitting for our models, is that we don't know what this surface looks like. We try some algorithms to sort of navigate this, and we try and work our way through. The observation is, you sort of get some of the insights here about what you need to do. So that you don't want to get stuck in a valley that may not be the lowest one, but you don't know where the lowest one is. So what that means is you're just sort of stuck with this notion of just trying a few different places. So you may want to start in a few different places on this surface. And you want a way of sort of exploring, so that you get, you explore the other valleys. Also, exploring more means looking at more values of the parameters. And so we get back to that whole concern about what is the computational time as we explore more and more values with more and more parameters. So gradient gets stuck on these ridges. And then, okay, that's sort of the observation here. So this is sort of a summary of the key things that you want to be aware of. So I showed you these two examples. This one, gee, if you can find it, that's great. Chances are it's not going to happen. This is probably a lot more common. So on this more common case, what do you got here in terms of concerns? So what are the things going to make it so that it's difficult for you to do parameter fitting? So what does the fitting surface look like? If I know where these ridges are, and if, for example, I know that, I can just be in this small area, I can sort of focus on that. But if I don't know that, I have a hard time. The number of parameters, okay, I'm showing this in 2 dimensions. Remember, even that simple glycolysis model had 16 parameters of note. So that's definitely going to make a difference. And then the other thing, of course, is where I start. Now, if I had the good fortune or maybe fantastic insight to start right on the hill, this final hill into the valley where that red dot is, that would be great. I could use gradient descent. I mean, at that point, it's just like this over here. But, how do I know that? Okay, so it's really what it comes down to is that parameter fitting is really a kind of exploratory data analysis. And much like you would do with exploratory studies to look for models, you sort of have to do some exploration as well with parameter fitting. There's a package that I wrote, something called SBstoat. That stoat is a kind of animal that sort of looks for holes and stuff. That's what's in SBI for systems biology. And this is a package I'll share with. It's available for free, but I'll do a little bit of work with that in one of the lectures I'm going to do in just a few minutes, and you'll get a feeling for that to sort of support this idea of exploratory data analysis. Let me pause for a second because I see a bunch of things in chat. Before I read things that have already been answered, what questions, Veronica or Lucian, need to be answered still?
Maybe if you could explain on the grid that was 64 by 64, that was on computational complexity, what the y-axis meant in terms of.
Sure. Yeah. Let me go back to that. Great question. All right. Yep. Okay. So the y-axis. Are we talking? Are we talking about? It's actually a 3-dimensional plot because I have 2.
Yeah. What are the
values,
settings?
Axes.
Think,
what does settings mean? Okay, so settings are the values assigned, the number of values assigned to parameters. So if I had a parameter like a kinetics constants K1, am I considering just 2 values? Maybe I want to consider 0.1 and 0.2. That would be 2 settings of the parameter. But how do I know it's just 0.1 or 0.2? Maybe I should have 8 settings. Maybe I should do 0.1, 0.2 all the way up to 0.8. That's the number of settings. And these are continuous values. So, I'm just sort of guessing about the number of settings I have to explore. The reason why I can look at the number of settings is each different setting to get an assessment of the RMSE, the residuals, sum of the squares. Each setting requires doing a simulation, and that's where we add up the time over here. Does this answer the question for, maybe the questioner can follow up and say, if that they need
I think in the discussion it's covered. So basically, you have a range of values that you think it might be in. You might select 64 evenly spaced. You might randomly select 16 of the values in there. You might have a variety of different search strategies, but
exactly, Lucian. Okay, great. Okay, so that's clear. Other questions that need to be addressed?
I guess ultimately the point is that you have to put in specific numbers in order to run a simulation. You can't say
exactly
X is between 5 and 6 and run your simulation. You have to say it's 5.5.
Exactly. And the way you figure out where between 5 and 6 it should be is you try a lot of values between 5 and 6. So, and each one you try requires another simulation. So, other questions?
I have a question.
Raised.
I am actually interested in your SBstoat model. In your project, what are the resources? Can you talk a little bit more about it?
Well, we'll actually do the third part of this talk is actually using SBstoat. So hopefully we'll answer the question at that point. By resources, what it is, it's a pip installable package. You say pip install SBstoat. So if by resources you mean computational resources, there's no server behind this. You're running it on your machine. All right, so but you can pip install it.
Yes,
does that
yes,
answer that
yes.
question?
Yes,
Okay, great.
it answers.
Okay, and we'll show that I'm in the process of converting it so that it uses read the docs documentation. I don't know if you're familiar with that. Right now, there's just a Jupyter notebook that gives examples of how to use it. Okay, other questions? All right. Okay, so what I'm going to do now, I'm going to go on to, this year we're doing a slightly different format. And so I have a little bit less time. So what I'm going to do is I'm going to go through in the shared drive. There are a number of different files, but the ones I'm going to go through first one, just sort of introducing some of the computations that you need to do parameter fitting. I'm primarily looking at the residuals calculations, and so that it involves enough in terms of Python usage. It's helpful, I think, to sort of run through an example there. And then we're going to talk about an example using SBstoat. There are some exercises too, if you care to do that. I probably will not go over them today. We'll see how the time goes. I saw some more things in chat. Is there more to answer?
There were just some questions about genetic algorithms, and I think those were generally answered.
Okay, great. Okay, good. All right, so I'm going to switch over now. I think I'm sharing my entire screen, so I think that this will work. But if for some reason, let me, good. Okay, so this should be it right here. Okay. And this is it. Great. Okay. So this is the file. Oh, let me go back. Actually, I'm going to start this right now because this will take a second. Let's see. This is the drive, the Google Drive. And here are all the files here. We just went through this one over here, the parameter fitting overview. And then that was the overview. This is the basics of the residual calculations, which conceptually sounds simple, but it's useful to go through a little bit of the detail. And then I'm going to jump over here to part 3, the detailed example. This one here, part 2, that's about looking at the details of the optimization algorithms themselves. Herbert and I teach a computational biology class where historically we have gone through a lot of detail on the optimization algorithms. I'm sort of coming to the conclusion at this point that there are really only 2 algorithms you would ever want to consider. Basically, there is gradient descent, which you want to consider, when you're at a point where you think it's a convex surface, and there's differential evolution, and all the other ones are not going to really help you that much. Any rate, I think there's probably a little less of it, but the idea behind optimization and what goes on with it is fundamental to doing the parameter fitting. And it's a fundamental technique for any sort of computational study. And I think this is just a graphic. Okay, so let me switch over here. And, oh, that's the wrong one. This is the right one. But I think I will run this anyway. And just so that we have that all set. Okay. All right, so you've gone through Jupyter Notebooks. I actually, and the Colab environment already. If anybody is not familiar with this, actually raise it right away because I could give a slight background. But this environment, this Colab environment, is really great for teaching purposes. It's not practical for something where you're doing serious studies, computational studies, because there are limits on the computation you can do. But for teaching and sharing of codes to show how codes work, it's wonderful. I make use of the table of contents a lot. It helps because you typically are developing a narrative through this, and sometimes you need to jump around. and that's helpful. There are two packages that we need to run this notebook. I usually do this conditionally. I also do this in a standalone environment, which is why I have this constant here, which I checked for. But in the standalone environment, I already have these installed. So obviously tellurium, which you spent a lot of time on, and then SBstoat, which we just made reference to. I recommend using the -Q option for the pip install because you get a lot less clutter on your screen. All right. What I've also done here, and right now what I'm trying to do is just give you a sense for what's going on here. There are a lot of sort of related code resources that are useful to have available. Many of them we've documented before in a GitHub repo. I don't know how many of you are familiar with GitHub. You don't have to really know anything about it. I'll just sort of explain where this comes from. So this is just this get shared codes over here is just a function that allows us to get at code resources that are stored on the internet in these GitHub repos. And then what you're seeing down below here are the codes that get included, just so that you have it, you can see what it's there, you can get a model. And then the last thing, which is very useful over here, is we're going to do some examples, some of which are, I don't think we do Wolf in this particular one, but this is sort of a general way of getting at these codes. But there are a few different data and model resources that are useful here. So we're going to mostly be working with this linear model, which I imagine you've encountered quite a bit. I'll show you that in just a second. This is going to be a, it's going to, actually you can see it right here. So this linear model is just it's a Python string, which is an antimony model. And you can see what it looks like. So we've got a path that goes from 1 to 5 with different species. You start with an initial value of 1, so all the masses with S1, and ends up at S5. And it's a simple model that's useful in terms of sort of understanding some of the basics of parameter fitting. And then these are all kinetics constants. In this baseline model, I set them all to 0 because we're going to do parameter fitting. This over here, this let me just look at this one right here. I'm not sure you've encountered data frames. It's there's a package called pandas that allows you to, let me just show you over here. It's a package called pandas that allows you to get sort of tabular format. So if I look at linear pathway, and you can actually just select it over here. They're very nice about having this. And then I run this. And then what it does is it gives me a table representation. So what this is a result of a simulation where I had assigned particular values to the kinetic constants over here. These K1 through K5, they're 0 over here. But in this, or I guess, yeah, they're slowly, K4 rather. And so by assigning those particular values, as it ran the simulation, I got these outputs. And so this is what a simulation output looks like. It has, and I'm sure you've seen this already in the tellurium discussion. So we have time is one column. We have then the values of each one of the floating species. And we get these, it depends obviously on the values of the kinetic constants. And not surprisingly, what happens, and this is over a period of, here's time is, that says, looks like it's time 1 there, and we go down to time 10. And we start out with all the mass up here with S1, because that's initially 10. And I guess we skipped from 0 to 1, I guess. Should start at, if we saw 0 in this particular data, it doesn't have that. And then we end up with, we should end up with all the mass then at the last one over here. I don't know why we have 11. I have to see what's going on there. That shouldn't be. You shouldn't end up with more mass than you started with. If you did, then I don't know, maybe that's an analogy to a perpetual motion machine. Any case, that's the format of these data, and that's what a data frame looks like. This is in contrast to what you probably have seen more of are NumPy formats here. So if I instead said linear, I got to spell it right, linear pathway. Okay, so this is an array, a NumPy array. If I looked at it as an array, it's not nearly as neat and that's why I tend to use data frames. The advantage of an array is that it's much more computationally efficient. Data frames tend to move memory around a lot. They're very flexible. They'll move memory around. That means that every time you do a computation with a data frame, it takes longer. And as we saw before, parameter fitting is very computationally extensive. So, maybe instead of 1 microsecond per simulation, now we're talking about 2 microseconds. Well, that doubles everything. So we would prefer the 1 microsecond. But even with the sort of, very careful coding and efficiencies, you can't overcome the fact of the computational complexity, because once you get computation times that are like at the age of the known universe, it doesn't matter whether it's half of the age of the unknown universe or a quarter of the age, it's still too much. You're not going to be able to do it. Anyway, so that's what these things are here, those in the background. Okay, let's talk a little bit of detail just to get you familiar with sort of computational environment here, much of which I'm sure you've encountered already from the tellurium lectures. Actually, before we start, I see a whole bunch of stuff in chat. Let me pause. Is there something I should answer?
It's mostly just a conversation about PyPesto.
Gotcha. Okay, gotcha.
We do have one new question.
Yes.
Distribution of data such as normality or non-normality for parameter estimation matter?
Oh, the distribution of, so I'm
the data.
Not sure I understand exactly. I'm going to interpret that question. If I've interpreted wrong, please let me know. So what I'm hearing is by the distribution, we're talking about distributions of parameters values. So that's that axis, the setting axis. If I quickly switch back here, I think I can do this because I have a, yeah, okay. So we talked about the setting axis over here. So how do we choose the values we're assigning to the parameters? We could choose it from a normal distribution. We could do some sort of grid search, something like that.
I think maybe the data. Maybe the data is talking about.
Oh, the data. Maybe the data.
Jalal, is it the data? Is it the experimental data you mean?
Well, I'm just asking about like, yeah. Sorry. If you like, you choose some data. So do you care like firstly about the normality of the data?
You mean experimental data?
Well, whatever like you choose. Yes. Yeah.
Right. Okay, very good. Thank you for the clarification. I guess we're not doing statistical tests here, at least in this discussion. And so distributional assumptions about the experimental data are less of a concern. I guess something that introduced bias, where we tended to get, values in certain regions, certain parts of the time evolution, maybe some experiments might, make, an observation lower than its true value in one part and higher in another. That would be problematic because we probably would fit the wrong parameter. But right now, we're not doing statistical inference. So that's less of a concern. Hopefully that answered the question. Although there are parts where statistical inference is done, and then obviously then distributional assumptions matter. Okay, so we just talked about the chemical pathway that's being studied. So this is going to be our running example here. It's a relatively simple example. And what I want to do is I'm just, again, focusing on one part, the calculation of the residuals. And this is because the residuals are so central to our analysis. And from that last question, actually, when people talk about normality, really what they ultimately going to want is that the residuals be normally distributed with a mean of 0. So that, somehow you've got this notion of being around a central value. All right, so I guess I did that here too with those plots. And then here you get to actually see this is the raw data that's there. So now we have a sense. And I actually, to underscore this notion about parameter fitting as an exploratory data analysis, the first rule of exploratory data analysis is look at your data. You have to understand what's going on. And one of the things that you see here right away, and I don't know if this came out, Herbert, in some of your discussions, but one of the things that's very interesting here is that we see that, and this goes back actually to the question that was asked before, to some degree about the distribution of the data. Okay, so this is going to be S1. We know it's S1 because it starts out with all the mass and then it goes down to 0. And the intermediate species, 2 through 4, have a similar thing. They peak, they basically peak earlier if they're earlier in that linear pathway. And where things end up is at S5. Now, what's very interesting here is this spread of values for S5. Now, remember, this is I think I had a modest, there was a modest amount of randomness that was introduced in the beginning for the data. But that randomness got amplified as we went through these various stages, because what happens is at each step in this linear pathway, there are delays. By adding some randomness to the values that are going through this pipeline, what we do is we essentially amplify the randomness so that by the end, when all the mass is appearing here and these guys are just zeros, this is where we see a lot of variability. And this will cause us problems with fitting, because we don't have a straight line, and so our residual sum of squares are going to be larger because we've got more variability. Hopefully that makes sense to folks. If there are any questions on that point, let me know, because that's actually an important consideration as you look at your experimental data and you're trying to do fitting. Obviously with variable data that has less variability, you'll get better fits. Okay. All right, so the fitting basics, we've gone through this before to some degree, but what we've got is we've got parameters that we want to be able to estimate. We're going to get observational data. The fitted data are going to come from the model, and then we're going to calculate the residuals. And so the key thing I'm focusing on here, and this is just to give you a feeling about how you calculate residuals. Okay, so this is the that analogy of that same algorithm I had before. Let's go through this exercise over here so you get a feeling for how this can actually work. So I did not mean to do that. Okay. So I guess in previous years, what we do is actually have people, go aside and run this. I don't know how you are right now in terms of being set up for environment. So I think it makes more sense for us to just work through this sort of together. And what I would like to do is, I know there are quite a few people here, but maybe we can make this a little bit interactive along the way. So we have what we're going to use for our data, our observed data is going to be this right here. So this is the array format for the data. And the reason why I want to use the array format, there are a couple of reasons here. One of them, and the primary motivation, is computational efficiency. The array structures in Python are much more efficient to use than the data frames. Data frames are nicer, they're easier, but this is much more efficient. I mean, we're talking like by a factor of 10 to 100. I mean, really big difference, you'll see. Okay, so that's our observational data. And then we've got this model, okay? And we want to calculate residuals because this is an essential part of the whole process. So, I'm going to do this residual calculation for one instance of what I call the settings or the parameter values. So we're going to assign parameter values. So this is something, let's go through this one, see if there's familiarity here. So what we've got is, here's the model. That's that linear pathway model. And you certainly are familiar with doing this, the loading, and then creating the Roadrunner object. Can someone tell me what's going on here? What's going on with these steps here, lines 3 through 6?
You're setting initial values?
Yes, right. So this is the way you assign values to the parameters. So, so this is a guess where we're saying, I'm going to use the values 1, 2, 3, 4 to see if those were the values that were used to generate the data. So this is our data up here. I want to see if those values are, how close they are to the values that were used for the data. So I'm going to assign values to this, and then I do this down here. What's going on here? Someone else want to offer?
you're simulating from 1 to 10 and taking 100 points.
Exactly. Exactly. So I've used those values, those guesses for the parameter values, and then I've got some fitted data. So now I've got fitted data, and I can look at it. Again, I highly recommend looking at it. Here's what it looks like. Now I want to calculate residuals. Okay. So
one quick comment, Joe. Just that
yes.
Earlier in the course, all we've done is just rr.k1 equals 1, which is just another way of doing it.
Yes,
this is
right.
a more generic way. So if you want to set like a concentration and put the brackets into your thing, rr quotes bracket k1 on bracket or something, you can set that way, but you don't.
I'll even write
other
it
ways
here.
of doing
Yeah,
it.
that's a good point. rr dot k1 equals 1.
Yeah,
it's
that's what we've been doing. Yeah. So.
Yeah. Okay, that's a good point. Thanks, Lucian. Yeah. Again, I don't know how much you've looked at this or from Python familiarity, but this behavior right here, where it's RR with a square brackets, that's how a Python dictionary works. You use those these square brackets around something in here. In this case, a string, and the Roadrunner instance will allow you to do that kind of assignment. So that's a very useful thing to have. Okay, so this is the next step here. So now we're going to calculate the residuals for S1. So we've got a lot of residuals here that we would like to calculate. We've got S1, S2, S3, S4, S5. It turns out that these data here contain values for all of them, S1 through S5. You may not be so lucky. You may only have S1 or S3, or you maybe have S1 and S5. So you may have to subset a bit and just choose which ones. So over here, can someone tell me what's going on here?
We haven't done slicing.
I'm sorry?
We haven't done slicing.
You just
you haven't done slicing. Okay, thank you. Go ahead.
You just get the first column, which refers to time, just extracting the time from that, right?
Right. Okay, fine. Thank you. And I thank you, Herbert, too, for slide. So this is a Python construct, something called slicing, that allows you, remember, we've got this table. And what this says is take the first part of this pair, this ordered pair, that colon there means take all the rows. So the first part indicates rows, the second part indicates columns. So this says take all rows, and then just Python begins indexing from 0, take that first column. So remember, time is the first column, you're taking all rows. So that's why you end up with times. And then over here, this is the observed values of S1. Well, S1 is the second column, and in Pythonese, that's the column index is 1. So again, we're doing the same trick over here. So now what we've got is observed S1 array is going to be this first column over here. All right, so now we have this is what the values look like. And now what we need to do is we've got, apparently, where did I do calculation? Okay, so you have to do a similar kind of thing for the observed values, slice it up like what we did with the fitted. So that's with the observed S1. And then we get the S1 residuals. And then we subtract one from the other. And with arrays, you can do this kind of thing. And this is what all the values are. And we'd want to do this with all of the observed, all of the fitted values for which there are corresponding observed values. And we'll end up with a whole bunch of them. And you see, this is what commonly residuals look like. There's a mix of positive and negative values. Some of them are large, some of them are small. You got to look at the exponents over here. I mean, some of these, you see small numbers. This one over here is a larger number, but see towards the end for S1, the residuals are very small. Someone want to make a comment about why the residuals are so small as we get to the end of the time course for S1?
Because there was so much variation.
Well, actually, let's go back over here. Maybe it'll make more sense. Okay, does it make more sense? One, we're looking at S1. And this is the fitted value. Actually, we should probably look at the original observation. Here's the observed values. S1 is the blue, this right here. Okay, what happens to S1 over time? You can answer this.
Decays to 0, so it becomes more precise.
Exactly, exactly. So in an absolute sense, it gets very small. In a relative sense, it might be big, but the absolute value of it is very small. How about if we were going to do S5, its residuals, what would happen?
The contrary, it will increase.
Exactly. And that's because look at all this variability over here in the larger values. So again, these are the things that, and remember what we're doing here is with this algorithm, the search algorithm, we're looking at the sum of squares of the residuals for all of the species we observed. So that means that over here, what's going to dominate?
S5.
Well, over here, it's going to be S1 because that's where the,
yes.
Yeah, right. Right.
At
But
the
over
end,
here, you're absolutely right. It's going to be S5. And in fact, overall, you're probably, and I think this is what you're answering. Overall, S5 is what's going to dominate because these are much more varied and much larger than what we've got over here. And this is what we have to be sort of aware of as we're doing parameter fitting, to be clear about what we're actually fitting to. Again, the residuals are at the core of all of this. And if we get our residuals screwed up or we're biased because of the data, then we're going to come up with screwy values for the parameters.
So if you took the log of those values and then fit to those in an absolute sense, would that help scale things a little bit better?
Okay, that's a great question, Lucian. And I have an answer for you, but I'm not going to answer it. I want to see if any of the students have an answer for that one. What do you think? Should we take the log?
I had a question about this. How about instead of calculating the absolute residual, calculating the relative residual for each of them? And
by relative, you mean like you're going to normalize relative to their true value? Okay, that's okay. So these are all. This is another example of kind of normalization. But can I get back to that one in just a second? Let me answer Lucian's question first.
Oh, sure.
Anybody? Do you have a question? I don't know. Maybe you have an answer to Lucian's question about taking the log. Okay, so the challenge with taking the log is we have 0 values here, or very close to 0 values. We're going to end up with huge negative values for log for these small guys over here. That's sort of a challenge. And so how do we do that? Then we're going to have scale differences. I mean, we could maybe take the logs of some and not of others, but now we're working in different scales. So that's a problem too. Let's go back to your question about the relative values. The challenge there with relative values is that now what's going to happen is we will accentuate small variations in this region, because these may have, maybe a difference of, 1 to the 10 to the minus 6, but relatively speaking, that may be big. So then we come up with big numbers there. I'm not saying that's the wrong thing to do, and I think at the end of the day, you have to look at your data, and if your data doesn't, to begin with, if your data doesn't have natural zeros in it, taking the log to stabilize variability is a great idea. And similarly, looking at relative differences, if you don't have a lot of variability, looking at relative differences is also probably a good idea. But you have to look at your data, understand what it means for you and your analysis. Is that a reasonable answer, or other questions?
I also, when I face this problem, and it's very there are many differences between different scales of one coordinate or another. I usually separate and fit one function in one part and another function in the other, taking the limits and then combine the two. It's very exhaustive, but it works always.
That's great. And I like that. I like that idea of, and you can separate in time, and you can also potentially separate across, grouping different chemical species, maybe treating those a little bit different. But absolutely, I mean, that's very reasonable. Again, it's sort of very specific to your data. And like you said, it's exhaustive, and it is very time consuming to try and figure it out. But it almost always requires some sort of thoughtful analysis like that to come up with something reasonable. And you know, this is not something that I talked about earlier on. I just spoke to the computational complexity, but this sort of, you know, careful attention to the nature of the data and sort of like, you know, some people call this statistical hygiene, you know, make sure you do this in a clean way. That's probably as important as all the algorithms, just to make sure you very carefully and cleanly handle the data to get the variances stabilized. Other questions or comments, observations from your experiences?
How about adding a regularization term to the optimization?
Absolutely. Okay, so let me explain to folks about regularization. Regularization is, and I don't know if this is what you mean. This is the definition I'm familiar with for regularization. Regularization to me means that in addition to finding the smallest value, like of the root mean square error, you also have a term in there that essentially gives you a penalty for having too many parameters. Is that what you mean?
That's exactly what it means.
Okay.
Yes.
Okay, fine.
To avoid
And
overfitting. That's, yeah, just to avoid
exactly,
overfitting.
to avoid
Yeah,
overfitting, right. Regularization, and so this really goes into the nature of the search algorithm that you're using. You know, it's a combination of optimization and search, and will you consider some parameters more than others? It's a great thing to do, but keep in mind that you still have this huge space you're looking over, but I think regularization can be very helpful. And it certainly, since this is an exploratory analysis, you should definitely consider that type of exploration. Other comments? Okay, let me move on. I'm going to go on to the last section, which is about actually using SBstoat. And let's see if I still have over here. Okay, so let me show you where I am. So now I'm giving you what I call detailed example. That's the one we're looking at. That's right over here. Let me move this up here and get it out of the way. Okay. And I think I'm still, okay. So this is a detailed example. I think what I've done here is I showed it for the linear model, and then we explore a little bit for the exercise, I think, is going to be with the Jenna Wolf model. So that, okay. And let's see here. Let me get rid of that for right now. And that, oh, I guess I've got a lot of these. Okay. All right, so usual stuff with the setup here, and then the helpers, and then what we'll come down to here is this is the data then that's being provided. You can see that right here. This is the end result. That's really what we'll play with. This right here is just so we can do plots easily. I usually put in some helper functions. Okay, once again, we have a similar, yeah, I think it's the same linear pathway, right? Okay, so we're running in the same boat, same context as before. Okay, so this is SBstoat, this package that I mentioned before. It's something that Herbert and I have developed. It is intended to integrate these notions of parameter fitting in the context of SBML models, and also help facilitate some of the exploratory data analysis that you need to understand what it is that's going on as you choose either different algorithms or understand the quality of the fits that are produced. So what you need, so what are the inputs to SBstoat? Well, you need the model, okay? We've talked about the observational data. And then here's a bit of a subtlety that we haven't really talked about, although we alluded to a little bit. So there, you know, certainly there may be a subset of the parameters that you're interested in, but beyond that, one of the subtleties, which, well, let me quickly switch over back to this guy over here. I'm going to go down a little bit. Yeah, let's look at this for a second. Okay. Okay. So one of the other subtleties about doing the search is it matters a lot where you start. So here's a surface that's clearly non-convex, you know, got a lot of hills and valleys. And as I mentioned before, if I started right here, you know, I could use gradient descent and it'd be real fast and I'd find the right parameter right away. But if I started over here, I mean, chances are I'm going to get lost somewhere in this region over here. I mean, it actually looks a little wider than when I get going that way. So where you start makes a big difference. The other thing that makes a big difference is how big of an area you're looking at. So here we've got, this is our range that we're considering for J2_k. You know, all these parameters, you have to specify to the algorithm where to start and what's the range of values I should consider. If I consider the values from 0 to 1,000, you know, which almost certainly would encompass what I wanted, then, you know, that's great. Everything is there, but I've got a huge search space. I'm going to run into this problem of it's going to take me much longer than the age of the universe. So, you know, I have to specify those two considerations. Obviously, the more narrow I get, the fewer parameters. So if I'm, you know, really smart somehow, and I say, I'm going to consider J2_k between 9.5 and 10, and this one over here between 0.95 and 1.05, I'm right in this area here. And maybe it doesn't matter so much where I start, because if I, you know, if I don't use gradient descent, if I use a lot of starting points, you know, I'll eventually get to that red dot. But if I said, let's start over here, you know, it's a small area, it won't take me long. Well, but I'm in the wrong place. So these are all things that you have to work out. It's always going to be a trade-off between, have I included what is the likely optimal value for the parameters, or have I chosen such a big area or a bad starting point so that it's going to take me longer than the age of the universe? No, those are the trade-offs. Okay, let's go back. Okay, so here I am. So when we say specification of value of the parameters, the subtlety here, oh, I guess I didn't mean that. Let me move away. The subtlety here is that you've got to specify a range and you got to specify initial values. And that actually is part of the exploration, is choosing your range and the initial values. You know, if you remember, which Indiana Jones was it? It was wasn't Temple of Doom, but something. I remember there's a scene where, you know, you have to drink from the chalice, right? You know, and they're like 100 chalices, you know, choose wisely. You know, and one guy chooses wrong and, you know, dies in a horrible way.
That's the Holy Grail.
So choose wisely. Okay, thank you. Holy Grail. I need to revisit my Indiana Jones movies. So so choose wisely. Think of that poor fellow who chose poorly. What happened to him? Okay, so the way SBstoat does this is that you have to start out by specifying what your parameters are that you're going to consider. So SBstoat has a parameter a parameter class where you say the name of the parameter, its lower value, lower end of the range, its starting value, and its upper end. And so for this, I just chose between 1 and 100, and we're starting in the middle. That's as good as anything. I don't remember what the value. Oh, I guess I could go back and look. I don't remember what the actual values of these things are. It's between 1 and 100, but you know, it's a reasonable range. For the data, you want to specify, so we're giving SBstoat an array. Array is not a data frame, so it doesn't have the columns identified. So we have to specify what those columns are in the array. And so that we end up with is we've got the model, which, you know, has the, oh, that's nice. It shows us actually what the model is over there. We're going to make this this array into something called a named time series. It looks sort of like a data frame, but it's more efficient. And then we list the parameters we're going to fit. And so that's this thing up here where we said this is the parameters we're going to fit. That's this whole consideration over there. So those are the inputs that we need. Then, as with, you know, very standard in these object-oriented frameworks like Python, is this does what's called, this is what's called a constructor. A constructor creates an object, is the Python terminology, which has certain characteristics. You can do things with objects. So we're going to create something called a fitter object, which is an example of one of this model fitter. So it's something that's going to be able to do the fitting with this information, the model, the observational data, and then the parameters we want to fit. And the way we do that fit is that The dot is something that is, now I'm going to do something with a property. Anything with a parens over here is a function. So there's a function here called fit model. And actually what's sort of nice about this notebook environment is, I believe if I do this right, I probably am not doing it right. Should be able to, I think, let's see here, let me try it like this. If I say, there we go, fitter, oh, fit. See, it'll tell me what it is I need to do. So fit model. So then I do that, I can do this. And now I know that I'm actually going to be able to run the fit. So let's, before we run that, this is something you can always do in Python, in the Jupyter Notebook. This is actually something you can run as a command, and then you can get more detail about that. It shows up in this help window over here. So this says that model fitter, this class, here are the things that you can pass it. So there's a lot of arguments that are available to it. This gives you an understanding about what it is that you might be able to specify. Okay, so let's actually run the model itself. So we run it for this particular one. It just finished running. And then what we can do is there's a report to tell us a little bit about what happened. Okay, so remember we had parameters K1 through K4. It looks like the parameters were probably assigned the values 1, 2, 3, 4. It just took a couple of seconds to do this, although it's a relatively simple model. There are a few things that are interesting here. So apparently the way we ran this, and you can do this differently, there's something down here called fitter methods. So fitter methods allows you to specify what search algorithm or optimization algorithm you're using. Apparently by default, it was using least squares. Probably one that's going to work in general better is going to be differential evolution, but it will probably take longer. But anyway, least squares seem to work okay with this one. We got values that are pretty close to what we would, the observational data. Here is the algorithm that was used. So function evaluations, this is a great one in terms of understanding the performance. Again, this is sort of exploratory data analysis. This says this is the number of simulations that run. That's that's the interpretation of functional evaluation. So it ran 6 simulations. Wow, that's pretty good. Run just 6 simulations, and then you find these values over here. And then you get some analysis of what it is that came out in terms of quality metrics. So this is let's see here. So this has statistical interpretation, actually the chi-square, and then there are other ones as well that give you some other insights. Generally, lower values of this is what it is you're looking for. It's good for comparisons, and you get some idea of correlations. So there's some details here that you will see in terms of how to interpret that. Now, I said that this was an exploratory data analysis, and so that means that you really want to look at the results and be able to see if this makes sense. On the surface of it, it looks like this probably did a good job, and we know that these values are probably pretty close, but it's good to be able to plot it out. So here's one example of, again, what's available through SBstoat. This idea of plot fit all fits that are happening. And so what it's doing here is the red line is the fitted value, and the dots here are the observed values from that observational data that we supplied. And you get a chance to see, if all of the floating species were fit well. Well, it looks like S1 was fit pretty well. S2, there's some things that went off a bit. S3, a bit more. S4, we see some things, a bit of more variability, and S5, quite a bit of variability. But, throughout, we're sort of towards the middle of the mess. So it's probably not too bad. And then you can see more of the details about the details of what that function actually looks like. Another thing that you can look at that's sometimes useful is looking at the residual values. So there's a whole methodology around looking at residuals. I'm not going to go through a lot of detail on that. You're, in addition to looking at, whether or not the residuals are close to zero. So here are residuals here. These are actually autocorrelations. What you're looking for is if you end up with autocorrelated residuals, then you have some sort of hidden structure or dependency in there that you may want to explore. So these are autocorrelations. So the lag is how distant the residual is from another residual. These horizontal lines here are essentially error bounds. So if you're within that, it probably is noise. If it goes outside of that, it's something to pay attention to. We see some of these, do go out a little bit, but most of them are within that. And the one at zero, at lag zero, that's, how well it's correlated with itself. So that should always be 1. That's expected. The other ones there, you hope have very low correlations. So anyway, that gives you a feeling for some of the stuff that's in here. So the next part I'm going to talk about here is a little bit of workflow about how we go about this. Let me pause for a second, and I see the chats have gone up. See if there's some questions to answer.
One question is just, you have data from a variety of sources, like you measure 5 different mice, and you get a bunch of numbers, but you think, maybe you think some parameters vary among the mice, but other parameters don't. So you would want sort of a global fit for one, but individual fits for the other ones or something.
Yeah, that's a great question. And adapting that is a challenge because presumably in your model, let's say you have 10 parameters and let's say 5 of them are global and 5 of them are mouse local, then how do you do those fits? That's a very interesting experimental design challenge, because obviously the 5 that are local, you probably don't want to fit them to, combined set of data. But for those that are global, you'd like to take advantage of the fact that they're global and pool data across mice.
Right.
So I'd be curious to know whoever posed that question. I can think of, like I said, just sort of like 30 seconds on this problem. I have a thought about how to do this, but I'm curious to hear what solution they came up with.
So hello, this is Meghna. So not necessarily a solution, but recently I got exposed to something called nonlinear mixed effects methods, where they take into account so if there is clinical data for multiple subjects, and then we have ODE models, the parameters tend to differ across various subjects. So I was thinking along those lines and was wondering if there is a NLME similar approach, which is probably used here as well. So just out of curiosity.
Well, it's a great question, Meghna. I don't know. I've never, to be honest, it's the first time I've heard of that particular challenge. The one thing that came to mind to me, and I'm sure, like I said, now I'm on like a minute on this problem, that might be useful is to fit, if you have 5 subjects, fit 5 subjects separately using for the global parameters combined data, and then for the local for each one of the separate ones. It's about all that you would have available. That's the one thought that came to mind. But this other thing, someone's probably had more than 5 minutes on this problem, has come up with something that probably is more sensible.
Thank you.
Oh, sure, Meghna. Anybody else? Any other questions or thoughts? Something about your research that maybe makes sense? Because the next step I'm going to do is try and go through a little bit more on the workflow that you do with the fitting.
In what situations it is certainly better to use differential evolution?
Oh, that's a great question. I would say that, here's my recommendation. This is actually, to me, sort of a simple answer. So what I would do is I would first try doing a gradient descent, see what I come up with. If I'm lucky, it might not be half bad because gradient descent is going to be fast. Then I would try differential evolution. Actually, what I would actually recommend is actually something Herbert does. I've seen him do. I would actually then recommend doing differential evolution, followed by you take the best parameters that come out of differential evolution and then do gradient descent. And the thought is that differential evolution might get you somewhere towards that final hill, you know, but not necessarily get you in the valley, because differential evolution doesn't work like that. But the gradient descent, what it'll do is it'll either leave you in the same place or close to it where you were before, or take you down a little bit. And so that actually would be my recommendation. The challenge is that differential evolution can take a while. And so you may need to do something that's sort of in stages somehow, just to see how much benefit you get from it.
I mean, I should add, I mean, we've tried quite a number of different fitting algorithms, including things like genetic algorithms, simulated annealing, all the gradient descent algorithms. And it was differential evolution that seems to always win. So I've never bothered, since then I've never bothered using anything else. Although it can be very problem dependent. So it's worth trying, you know, other ones anyway. Once you find one that works, then you use it on the model. I mean, our problem is we have to do these fittings thousands and thousands of times. And you can imagine if each one, I mean, this one that Joe did took a couple of seconds, but if the model's bigger, it could take hours and hours to fit a single one. And if you've got to do 1,000 of those, you can imagine how long it's going to take. So it's important you pick the right optimizer. So yeah, I think you want to explore it first, find out which optimizer seems to work best with the data and model you have, and then scale it up.
There are also things you can do with like multiple searches at the same time with like different initial values.
Yeah.
You can do like heating. It's sometimes called like you basically sort of take your landscape and sort of smush it, so you're more likely to sort of bounce around, and then like simulated annealing sort of like makes everything flat, and then slowly you get your mountains and valleys back as things
I mean, simulated is not a bad one, actually.
And you can use these things in concert with each other too, if you can find. Certainly, if you're a good enough programmer, you can just sort of like program it yourself. But there's things out there that.
The other thing that Lucien mentioned, multiple starts. I definitely recommending multiple restarts. So you may find a solution, but I would do this like 10 times, maybe. I mean, Mike Koshin has done. He knows more about this than I do, but you restart it multiple times to make sure you get the same solution, and then you're confident that it's probably found the right solution, the minimum.
Yeah, especially if you've started from different initial conditions.
Yeah, definitely.
Great. Any other questions on this or anything else, any other material that's been covered so far? Okay, so I'm going to go for about probably another 10 minutes max, just to leave time at the end. We're also, you know, towards the end of, you know, 4-hour segment, or the second of second 2-hour segment. So I imagine with many different time zones, so I imagine it's getting sort of late for folks. Okay, so remember the challenges, or, you know, some of the considerations from before, you know, the nature of the fitting surface, how the algorithm is exploring, and where you're starting, and the scope of what you're searching. I think both Lucien and Herbert mentioned the starting points, but the scope of what you're searching also makes a big difference. If you look at, for example, the Jenna Wolf model, actually, let me bring that back because that's very relevant right now. Let me go back to the beginning here where you can see the values. Here we go. Look at the difference in scale of these parameters. So this is what she used in her model. And you see things that are basically single digits up to, you know, 50,000 plus. And if you're presented with this model and you don't know a priori what scopes to use, do I go from 0 to 1 million? And, you know, so at that point, it's really hard to, you know, figure out, you know, what's a reasonable search. If I'm doing a grid search from 0 to the a million for 16 parameters, it's hopeless. That's never gonna help, unless it's convex. If it's convex, great, but that's not going to be the case. It's not going to be convex.
I feel like searching log space can also help you here, because then you don't have to. You're sort of like checking different magnitudes instead of just checking different.
Depends how fine-grained the surface is, though.
Yeah, exactly. I mean, because Lucian,
Yeah.
you've just changed the units, but you haven't changed the underlying characteristics.
Right.
So
it
now,
depends
you know,
on what the surface of that you're searching is.
Yeah, ultimately it depends on the surface, right? And we don't know the surface, and it's in very high dimensions that we can't even visualize.
But
so this
like
is,
as an example, I would imagine that, like, I don't know, J5K is 80 here. Like the numbers around 80 are probably good. The numbers around 1 are probably terrible. The numbers around 100, around 2000 are probably terrible. If you did a log search, it would zoom you into the 80 magnitude. And then you would switch with different bounds once you sort of found it there or something. That's a guess. I don't know.
Maybe.
Yeah, not to dampen your optimism, but part of the problem is that there are interactions between the parameters.
That's also true. Yeah.
Yeah, and so that's what makes this truly a combinatoric problem, and why it's such a massive computational challenge. But okay, that's where, you know, as you can, I don't want to discourage all you folks out there, but I mean, it goes back to why it's so necessary not to do this blindly. Use every source of insight and analysis you can do to sort of tease apart what makes sense, especially in combination.
Yeah, and someone in the chat mentions like doing mathematical analysis first to try to like minimize the number of variables you're searching, or just
absolutely. And that's a great strategy. I mean, you can tell one of the key things here that makes this complex. I mean, you know, that's what this chart, I'll go back to this one more time. So what this chart here was all about was to point out the primary considerations are the number of parameters and the number of different values you can consider for the parameters. If you can reduce the number of parameters, go from this red zone over here to something that's, you know, closer to a blue, much better, you know. And so if you could have some sort of a strategy where you start out with a few parameters that you could explore in great depth, then and then expand a little bit where these parameters are pretty much set, and then you can go on to something that's more. So over here, you may be exploring 16 parameters, but actually 8 of them are already specified. So instead of doing 16, you're actually doing 8. You know, that's the kind of thing, like I said, the strategies you use will vary depending on your problem, but these are the fundamentals. It's the surface, it's the parameters, it's the number of settings, it's where you start. So it's all those things in combination. Okay, so down to the finish line here. So here's what you probably want to do, or here's one thought about this, is to somehow select a subset of the data and focus on that. Then fit the model to that, and then be able to analyze how well your parameters work in that set, and then generalize from there. But there are many other variations on this that you might do. And in this last section over here, so for this simple workflow, I typically, you know, something that I can comment on, because the strategies I think are going to vary a lot depending on the problem at hand you have, the number of parameters and so on, what you can infer about reasonable settings and starting points. But one thing I will emphasize computationally as you're doing sort of the mechanics of this analysis is, as much as possible, encapsulate your process, your search process, into functions. And that's what's done here. This do fit function is something that tries to encapsulate a process here where it's considering a number of different columns, for example, you might fit, or n times that you might want to consider. And if that's my process, I have it in a function so that then I can rapidly look at the outputs here. So I've got the fitting report. In this case, I'm looking at the simple one. This is least squares, so it's pretty trivial. But I probably want to look at it something like this fitting report and this plot. And if I can look at these two things, maybe I can make quick judgments about what's going on and how I need to make changes. So again, I highly recommend that type of structure to it. What I was going to do is just scroll on down a little bit more. This is changes for the optimization algorithms you might use. This is an example of looking with differential evolution. Okay, and this is going through, as you go through this part of the notebook, it goes through some detail about each one of these challenges, like what's the start of the search and what's the scope, and how you might then adjust your fitting to be able to consider all this. Now, Okay, what I was going to do now, just briefly, is give you a feeling so that if you wanted to do sort of like a controlled exercise, maybe you're at the point right now where you're going to embark on some sort of computational study, but you're not there yet. Oh, before I do that, let me just point out something else. So here's an example of where I'm not going to even focus on what the input was, just the output. Here's an example of where some choices were made to do an exploration or setting of parameters that didn't work so well. So you can see, here's an example of where the fitted value, the line, works well for S1, less well for S2, and gets worse and worse as we go along. And it's apparent a little bit, I mean, you'll see this in the chi-square. I think our chi-squares before were triple digits. Now, or I think it was, yeah, it was like in the hundreds, and now it's, you know, in the thousands. So you can see it that way, but it's more graphical to also look at the picture as well. It'll give you more insight about where the problems are. So this exercise, which you may want to explore depending where you are, is basically to repeat this process that we had above for the linear pathway. Do this for the wolf model. And it's going to be a little bit more involved because there are more parameters. You may want to consider a subset of the parameters, but you have the data available to you. You have the techniques available to you. And this will make it so that it's very easy when someone shows you something to sort of passively think, aha, this makes sense. It's very different when you try and do it yourself. And so this gives you some sort of controlled environment. We could just extend this notebook and then be able to see if you've really absorbed the concepts about what's involved with fitting. Questions at this point? Discussions? Okay. All right. Well, if there are no questions at this point, I think what I'll do, since we have a couple more minutes, let me go back and I can explore a little bit with you some of the more advanced ideas in the in this other notebook. This is the one, number 2, the optimizations, and give you that next level of detail for those who care. We have about 10 minutes maybe, and we can just sort of see how far we get. I'll run the whole thing and then sort of talk my way through it as we're going through. So this is you know, set up in a similar way. In this notebook, the only dependency is tellurium, because we're looking at the fitting algorithms or the external dependency. I mean, there are other ones here. But that's the main external dependency. Okay, so let's go over here to the optimizations essentials. It gives you a sense of framing about, we talked a little bit, you know, Herbert and Lucian were talking about some of these algorithms. We talked about gradient descent. We talked about differential evolution. Lucian made reference to something called simulated annealing, which is another kind of optimization algorithm. So all these algorithms are doing something similar. It's a way of searching for the best value to a function. It's going to be you know, you have some function that's a function of many variables. So our variables in this case are parameters. And they, you know, we want to know what values to assign to these parameters. So here's this function f with, you know, a bunch of parameters that are there. So we're going to use a bolded X to indicate this vector. Each one is a parameter. That's how many parameters there are. And the best one is X star. So that's what we're searching for. And what we have in the way of our optimization criteria is this residual square of the results. So what you see here are examples of these fitting surfaces, and it includes the plots to do it. So you may enjoy exploring this a little bit, much like I did with those 2 fitting surfaces before, because you had the actual code there. There's some discussion here. This is what we're talking about before with convex curves. You get a sense for that. So remember, convex surfaces. So this is a surface, this is a two-dimensional surface. What I was showing you before were three-dimensional surfaces. So this one here, here's a curve which has a value that varies according to, the value is the y-axis, and x is the parameter that's what value it's set to. And the thing about a convex surface is it always has this bowl shape where it comes down to the bottom here. And if you just follow the slope down, you'll always get to the bottom. And that's what makes convex surfaces so great. There's also this notion about the local minima is the global minima. So if I'm right here and I say, gee, if I move here, am I smaller? That's great. But it's not the local minima because I can keep on going down. It's only one at the very bottom. And then you can get non-convex curves too. This is a more elaborate, not obviously those are the more common ones, are the non-convex curves. So here's an example of doing the kind of calculations to invoke these search algorithms. It uses a package, a Python package called in the scientific programming part of Python called Optimize. And it gives you examples about how to program this. This is a little bit more involved programming because it includes the idea of actually one of your arguments, instead of being data or a constant, one of your arguments is actually going to be a function, because the package has to know what to do about, what function actually gets optimized. And that's part of what you'll see in this notebook. It's a description about how to do that. And then down here, it gives you some opportunities to explore this further. So hopefully this will give you an idea of if you wanted to explore your own ideas for how do you do optimization. Hopefully this gives you the background and context to do it. All right, I think I'm about talked out at this point. So I see we have a few more things in chat. Is there something we should discuss?
Things in chat were congratulations. It was interesting.
Yeah.
Okay. All right. Well, that's good. I'll have to print it out and frame it. Good.
There you go.
Okay. Yeah, this is, the challenge with this material is there is a lot of this. If a lot of information is available online, but when you read it, it's incredibly dry. And very technical. And so it's difficult to plow your way through. And I know something that, as I mentioned before, Herbert and I teach a computational biology class, and this material is an important part of that class. And one of the things we've tried to do is make this more accessible. I think the BioE students are very mathematically capable, but this requires sort of a level of intuition that you have to combine with sort of the mathematical, technical part. That requires a bit more sophistication and some skills that you have to develop as well.
Is there some written material that you would come across that you would recommend, or have you written any written material that you can point to?
I haven't.
My textbook.
Okay, there you go.
Is your textbook has this in it?
Yeah, there's a whole lot of this stuff in there. So I have a textbook, systems biology textbook, and there's three big chapters in there. One is on optimization algorithms, and one is on fitting experiments and what happens when you leave out data, what happens to your confidence in the parameters you fitted. And then there's a whole chapter on Bayesian inference as well. So
I remember at one point you had to link sort of directly to your own website where you could.
Yeah, better to direct it to the Amazon site, I suppose. If you just let me see. Let me pathway modeling. Let's see if it's the green one. Oh, there it is. So I'll just put it in the chat. So there's three big chapters in there. I try to explain everything in English. But you can get the PDFs for this as well, actually pretty cheap too. Where's that? That's in Gumroad. You get that. Let's see. Where's search? What do I discover? Here we go. Hold on. Oh, I can't find it.
So I would appreciate, while Herbert's looking for this, I would appreciate feedback about how the session went. If there's any discussion, I mean, you could put it in chat, you can send me an email or discuss it now. Things that you felt, maybe spent too much time on, things that I didn't spend enough time on. I'm sure Herbert, if you haven't given him feedback, he'd love to have it too. So any discussion, we just have like a minute or more in this session. Because you've invested quite a bit of time, 4 hours, and for some of you, it's very late or very early. So,
yeah.
Pedro just said that it would be good to have exercises, which if you look in the folder, Joe does have them. And I just wanted to say that we'll have some extra time on Thursday in the second half of the session. So hopefully people, if you want to review some of the exercises in the meantime and go through them on Thursday, we can do that too.
okay.
Yeah. Oh yeah, here's the PDF. Here we go. And this is up to, it says 2020, but actually it's revised in 2021.
So there was one question about some standard literature to look on this. I'm not sure. I think Herbert's pointing you to his book, which I agree. I think it's one of the few places where you get sort of a practical guide. I'm not sure what else. Okay. Yeah. Okay. I mean, that's what, and I mean, there are other practical guides, but not specifically for biological modeling. There's a whole literature on regression diagnostics, for example, which is very practical stuff, but it's not going to take you through, some of the issues with biological models. Okay.
I mean, there are lots of reviews, but again, they don't go into practice much, which is the problem.
On a different tack, we have another question about just how to easily search the literature for numbers for your models that may have someone may have measured and stuff like that. There's mentioned SavioRK. Are there other online relevant resources? Are the relevant?
Doesn't biomodels have something now, a resource for parameters? I think they have some research, and there are also the models themselves that you can look at.
I mean, I think Veronica is going to say something about this later in the week.
That's good. Because I mean, that's a great thing to do, because obviously, even if it's not, the correct physiological value for your model, if it gets you closer, a closer starting point, you saw that that's one of the critical considerations. Am I closer to the, a correct value, or maybe it restricts the range. Maybe there are physiological limits on, enzyme catalysis that let you know that it can't be higher than a certain value. That's also helpful.
But in general, the process is like you find a paper, you see the model there, and you try to reproduce computationally, right?
Yeah.
Right.
Is the natural process.
Exactly. And that's, as you know, my background is computer science. This is so analogous to how people write programs. You find an example of something you want to do, you see if you can do it, and then you extend it from there. Same thing with biological models.
Yeah. At most, if you are building a new model, and in my opinion, you are not so constrained to find research with the real values. If you're building a model, you are trying to prove a point. You set parameters that work and show the model working there.
Right.
Oh, these are more conceptual models. Is that the idea?
Yes.
Yeah.
Yes.
Yeah. Right.
For example, you want to prove that you need this extra equation for it to work, etc. So
yeah,
this type of hypothesis.
Okay. Yeah.
Yeah, and another variation on that is you might make a statement about what are the kinds of kinetics you have. So not necessarily including another reaction, but you're saying with this kinetics, it works, and with this kinetics, it doesn't.
Yeah.
Okay, I think we're about done. Feel free to follow up. I mean, I'll be chatting with you tomorrow too. I guess it's verification and validation. So